As you will glean from my position in the lineup today, I'm also a basic scientist. And in my lab, we study mainly the effects of estrogens in the adult brain. And at first pass, maybe it wouldn't seem so surprising that there could be sex differences in the effects of estrogens in the brain, because we typically think of estrogens as being hormones that regulate reproductive physiology. And so it's really important for me to be clear at the outset that I'm going to be talking about estrogens in a very different context, not as hormones, but rather as neurosteroids, steroids that are synthesized directly within the brain and independently of the peripheral endocrine system. And the idea is that neurosteroid estrogens are synthesized in the brain, where they act locally and rapidly on a timescale of just a few minutes to modulate various aspects of neurophysiology, including synaptic transmission. And we know that neurosteroid estrogens are synthesized in the brains of both males and females, and that they can acutely modulate neurophysiology in both males and females. And we got started doing this work for a variety of reasons, studying female animals, female rats. And that was fine for a few years, until we began to get results, both with estrogens and also with non-estrogen manipulations, that just did not match the published literature. And it began to dawn on us that one of the reasons that our results might not match the published literature is that we were using females, whereas most of that literature had been generated using male animals. And so what I would like to tell you about today is some of the discoveries we've made by comparing mechanisms of synaptic modulation directly in males and females. And I'll tell you about two lines of work, just a little bit about two lines of work in the lab in which we've found some interesting sex differences in mechanisms of synaptic modulation. Before I get into the details of our experiments, though, I think it's useful to put the results in some context by taking a step back and thinking about sex differences in the adult brain more generally, okay? And when we do that, we can divide sex differences really into two broad classes: quantitative sex differences and qualitative sex differences. And when we think about quantitative sex differences, those are cases in which each sex has or does something, but one sex has or does more of that thing than the other sex. So you might be interested in a particular parameter of brain function, and you might measure that among many individuals of one sex and obtain a distribution of that measure in that sex. And then when you make the same measurement in the other sex, you might see a shift in that distribution. And the difference between the means of those two distributions would reflect a quantitative sex difference. And there have been many of these discovered. I think if we look for them, we will find many more. And of course, depending upon what the parameters are that you're measuring and the magnitude of those differences, some of them will probably turn out to be quite important. Now, distinct from these quantitative sex differences would be qualitative sex differences. And I want to highlight two types of those. The most obvious one would be sex-specific effects, okay? And in a sex-specific effect, one sex has or does something that the other sex does not have or do. So you might measure a distribution of some parameter in members of one sex, but when you measure the same thing in the other sex, you find that all of the individuals are at zero for that measure. And I'll actually show you at the end of my talk a couple of examples of sex-specific effects. Okay. Now, the other type of qualitative sex difference that I want to mention, which I think is maybe biologically even a little bit more interesting and certainly trickier to study, are latent sex differences. In this case, each sex has or does the same thing, but achieves that through distinct underlying mechanisms. Okay? And in this way, you might present a stimulus to members of each sex, achieve the same endpoint in both sexes, but as you explore the mechanisms by which that endpoint is achieved, you find that each sex reaches that endpoint in a different way. And I'll show you an example of one of those as well. Okay? Now, the experiments that we do are focused largely in the hippocampus, the adult hippocampus of rats and mice. The experiments I'll tell you about today were all done in rats. And so what I'm showing you here is a parasagittal section of a rat brain with the hippocampus blown up to the right. The principal cells in the hippocampus are pyramidal cells, an example of which is shown here, and that I'm going to diagram like this, where, let's see if I can get this cursor to work. So this would be the cell body of the neuron, and the thick lines are the dendrites, and then the thin line with an arrow there is intended to be the axon. And of course, like other neurons in the brain, these cells receive both excitatory and inhibitory synaptic inputs. The inhibitory synapses use GABA as a neurotransmitter, and inhibitory synapses are actually formed all over these cells, but they're particularly concentrated around the cell body. And they also receive excitatory synapses that use glutamate as a neurotransmitter, and those glutamatergic inputs are formed mainly in the dendrites of the cell. And the latent sex difference that I'll tell you about today is in acute estradiol modulation of the excitatory glutamatergic synapses in the dendrites, and the sex-specific effects that I'll show you today are related to modulation of inhibitory synapses at the soma. Okay, so let's start off first thinking about a latent sex difference in the mechanisms by which the steroid estradiol acutely potentiates excitatory synapses in the hippocampus. And our interest in this really stemmed from our efforts to understand a really fascinating result that was published by Tim Tyler more than 35 years ago now. Okay, and what he did was he used extracellular field potential recording in slices from adult male and female rats, and he applied estradiol to those slices and found that in both sexes, estradiol rapidly potentiated field EPSPs on the order of about 5 to 10 minutes. Okay? And as the evidence for neurosteroid estrogens began to accumulate, more and more people got interested in trying to understand how estrogens might act to rapidly modulate neurophysiology and synaptic transmission in the brain, my lab among them. And we were particularly interested in trying to determine the relative contributions of modulation of excitatory synapses and inhibitory synapses to this phenomenon. And so I want to show you first how we look at excitatory synaptic transmission in our experiments. Okay? If we go back to our picture of a dendrite, as Peg mentioned, these are sites of excitatory synaptic input to neurons. In fact, an excitatory synapse is formed on the head of each of these little dendritic spines that densely stud the dendrites. And we can assay the function of these excitatory synapses using electrophysiological recording. In our case, of individual hippocampal pyramidal cells. And when we're interested in studying excitatory synapses, we include drugs in the bath of a hippocampal slice to block inhibitory synaptic transmission. And we place a stimulating electrode in the dendritic layer to activate these excitatory synaptic inputs. And we can then record glutamatergic excitatory postsynaptic currents. And so the experiments that I'm going to tell you about next are those in which we are measuring EPSCs. And so one of the first things that we did was to ask, how does estradiol application to hippocampal slices affect EPSCs? And I'll show you the results first for females. Okay. And this is an example of one of our experiments. This would be a representative recording from one single female hippocampal neuron. On the left, you see 3 overlaid traces from different time points in the experiment. And in the middle there, you see the time course of the change in EPSC amplitude throughout the experiment. There's a baseline period, and then that bar with E2 above it is 10 minutes during which we applied estradiol to the slice. And you can see that in this cell, estradiol rapidly increased EPSC amplitude or potentiated excitatory synapses. That's sustained even when we wash estradiol off of the slice. And then we terminate the experiment by applying a glutamate receptor blocker, an AMPA blocker, DNQX, at the end of the experiment. Okay? So almost all the data that I'm going to show you today are laid out like this, so it's important that everybody understands what they're looking at here. So we see that estradiol potentiates excitatory synaptic transmission, but that occurs only in a subset of cells. Not every experiment shows this result, and you can see that when you look at data from many cells plotted together, where I'm showing you the average EPSC amplitude before estradiol and after estradiol, And the colored points are those in which we saw a statistically significant change in EPSC amplitude, okay? So you can see that there's a mixture of those with a change and no change, and you can see that better if I take the same data and plot them normalized to initial EPSC amplitude, and you see that in females, in 9 out of 16 cells in this case, we see an increase in EPSC amplitude of about 86%, okay? So we see a potentiation of excitatory synapses in just over half of our experiments. So what happens if we do the same experiment in males? What's the result? Okay, so we do exactly the same experiment. We apply estradiol to hippocampal slices for males. We get exactly the same result, okay? In this case, in 11 out of 18 cells, essentially the same proportion, we see an increase of about 86%, exactly the same as we do in slices from females. And so anybody would think that there is no sex difference in the acute estradiol potentiation of excitatory synaptic transmission. It looks exactly the same in males and females. But that would be misleading because there is a sex difference. And that difference has to do with the mechanisms by which estradiol potentiates excitatory synapses. So to explain that to you, I need to tell you a little bit about how these rapid estrogen effects work. Okay? It turns out that in addition to their classical role as nuclear transcription factor receptors, a subset of those same estrogen receptor proteins, the two forms, estrogen receptor alpha and beta, are trafficked to membranes, including the plasma membrane, in a process that depends upon cavelins, which is that little purple thing there, and also lipid modifications of the estrogen receptor that tether it to the membrane. And estradiol binding to these membrane-associated estrogen receptors stimulates interactions between the estrogen receptor and integral membrane proteins, including growth factor receptors and also G-protein coupled receptors, to rapidly activate a wide variety of downstream signaling cascades. Okay? And most of the acute effects of estrogens that we know about in brain areas like the hippocampus operate through a mechanism like this, all right? And so what I want to show you are just one experiment, really, from a series in which we have tested the roles of different kinases in this estradiol-induced synaptic potentiation. And in fact, other labs have done this as well, but interestingly, only in one sex or the other sex, and never actually comparing the two. And I'm going to focus in on our experiments on cyclic AMP-regulated protein kinase, or pKa, as an exemplar of multiple sex differences that we found in the mechanism by which this occurs. Okay? So in this experiment, we're going to ask, how does an inhibitor of protein kinase A affect estradiol-induced excitatory synaptic potentiation? And again, I'll show you the data first from the females. Okay? Here are the results of a typical experiment. Okay? PKI is the pKa inhibitor that we use. So you see that itself does not have any effect on synaptic transmission. But when we apply estradiol in the presence of PKI, we see no change. But then, of course, because about half of our experiments show no effect of estradiol anyway, we need to wash both off the slice, apply estradiol a second time, and in this case, we see the increase in EPSC amplitude, indicating potentiation of excitatory synapses. Okay? So here are group data from experiments like this, showing that in each of the estradiol-responsive experiments, inhibition of pKa using pKi blocks that effect. Okay, now, just to be sure, we wanted to do the same experiment with a different pKa inhibitor, in this case H89, and we see exactly the same result, that inhibiting pKa blocks the effect of estradiol to potentiate excitatory synapses in females. Okay, so everybody has this in their mind. Now the question is, what happens when we do the same experiment in males? Well, we get a very different result. We find that in males, inhibiting pKa fails to block estradiol-induced synaptic potentiation. This is one representative experiment. Here are the group data. Okay? In all of the experiments that we did, inhibiting pKa with pKi failed to block this effect. And of course, we did our experiments with H89, and we get the same result. We even tried higher concentrations of H89, and in every case, we still saw plenty of estradiol-induced synaptic potentiation. Okay? And so you can see this effect better if I put all of those summary data together when it's very clear that in females inhibiting pKa blocks this effect, and in males it fails to. And this actually reflects then a version of a latent sex difference in the mechanisms that underlie this apparently identical effect of estradiol to acutely potentiate synapses. And really this is just one of multiple sex differences that we found in this effect, ranging from the constellation of estrogen receptors that are involved to the way calcium is handled intracellularly and the different signaling mechanisms that act downstream of that extranuclear estrogen receptor activation to produce apparently the same effect in males and females. Okay? And I show this result really to make the more general point that males and females can and do differ in some molecular mechanisms of synaptic modulation in brain areas like the hippocampus. And I think that is important mainly in the context of the development of therapeutics, right? Drugs that are intended to target a specific molecular pathway in the brain and that may in fact have distinct downstream consequences in males and females. And so I think it actually, as basic neuroscientists, we really owe it to our translational and clinical colleagues to determine whether the types of basic molecular mechanisms that we identify in animals are true in one sex, are true in the other sex, or are true in both sexes, so that they can use that information in the design and the interpretation of any translational or clinical studies that may be derived from this kind of basic science information. Okay? Okay, so let me shift now and talk about our evidence for sex-specific mechanisms, in this case, mechanisms by which estradiol acutely suppresses inhibitory synapses. Okay. In these experiments, we're going to use the same type of approach, that is whole cell voltage clamp recording of individual hippocampal pyramidal cells, but now we're going to be focusing on those GABAergic inputs that are concentrated around the cell body of each neuron. So in these experiments, we're going to include drugs in the bath to block glutamatergic synaptic transmission, and we're going to place our stimulating electrode in the cell body layer to activate those perisomatic inhibitory inputs, and we're going to record GABAergic inhibitory postsynaptic currents, or iPSCs. Right? And so, again, the same experimental design. We're now going to ask how estradiol application to slices affects now inhibitory synapses, and what we find is that conversely to the effect of estradiol on excitatory synaptic transmission, a 10-minute application of estradiol acutely suppresses inhibitory synaptic transmission. Again, that suppression is sustained even when we wash estradiol off of the slice, and we terminate each experiment by adding a GABA-A receptor blocker at the end, and that's what that SR is. Okay? Now, similar to the effects of estradiol on excitatory synapses, we also see acute modulation of inhibitory synapses in only a subset of experiments. In this case, the plot shows you only the estradiol responsive experiments. When recording iPSCs in this configuration, we see the effect in just over half of the cells, in this case 6 out of 11, and the decrease averages about 44%. Okay? So what happens when we do the same experiment in males? Okay? Test how estradiol application to male hippocampal slices affects inhibitory synaptic transmission, and we see nothing. Okay? We see absolutely no evidence of acute estradiol modulation of inhibitory synaptic transmission. This plot shows you that in 22 different experiments, we saw no modulation. We've tried multiple concentrations of estradiol. We've tried hippocampal slices from gonadally intact males or from castrated males, and we see no evidence of this. Okay. So in this case, this is an example of a sex-specific mechanism of synaptic modulation, something that occurs in the female hippocampus, but not in the male hippocampus. Okay. Now, I'm not going to take the time to go through all of the details of the molecular mechanisms by which this occurs, because those data have been published, but I'm going to summarize for you how this works by going through a schematic. Okay? And so we have shown that this suppression of inhibitory synaptic transmission works specifically through an interaction between the alpha form of the estrogen receptor, ER-alpha, and mGlu-R1, metabotropic glutamate receptor 1, in the plasma membrane of the postsynaptic cell. And of course, mGlu-R1 is known to activate phospholipase C, which we've shown to be required for this effect. And phospholipase C hydrolyzes phosphatidylinositol bisphosphate into diacylglycerol and inositol triphosphate, IP3. And we've shown that it's the IP3 arm, not the DAG arm, that's critical for this. IP3 activates the IP3 receptor, which is located on the endoplasmic reticulum. That liberates calcium from the endoplasmic reticulum, and in this case acts through a series of steps that are not completely defined, I'll admit, to stimulate the synthesis of a key endocannabinoid, anandamide. And that anandamide is released from postsynaptic cells. It acts on CB1 receptors located on presynaptic inhibitory inputs to suppress the release of GABA from those inhibitory inputs, and that is how estradiol acutely suppresses inhibitory synaptic transmission. Okay? And we have explicitly investigated the possibility of sex differences in the interaction between ER-alpha and mGlu-R1, in the ability of estradiol to stimulate an increase in IP3 levels in the hippocampus, and in the ability of estradiol to stimulate the release of the endocannabinoid anandamide. And what we found is that the critical sex difference in this case is in the interaction between ER-alpha and mGlu-R1. And we know that because we can sort of leapfrog over this interaction and target other steps downstream in the pathway, and we get the same outcome in males and females. So the key step here is in the ability of estradiol to promote an interaction between membrane-associated ER-alpha and mGlu-R1, right? Now, there's one last experiment in the series that I want to tell you guys about, because it's actually the first experiment that we did that got me started into thinking that there were likely to be sex differences in these patterns of synaptic modulation, okay? And that comes from our manipulation of enzymes that metabolize anandamide, the endocannabinoid, okay? So anandamide is degraded by an enzyme called fatty acid amide hydrolase, or FAAH, all right? And in the course of working out the mechanisms by which inhibitory synapses are acutely modulated by estradiol, we did a series of experiments in which we applied a FAAH inhibitor to hippocampal slices. Okay, and I'm going to show you the results there. Okay, so in this case, we used an inhibitor called URB597, okay, and we asked how does it affect inhibitory postsynaptic currents in females, and these are the results that we got. So this is independent of anything to do with estradiol. This is just applying the FAAH inhibitor, and what we found is that in more than half of the experiments we did, only the responsive experiments are shown here, applying a FAAH inhibitor itself decreased iPSC amplitude in the hippocampus of females, okay? And that actually is sort of important to interpret in that it shows that there is tonic anandamide release within the slice. So anandamide is being released, and if you block its breakdown, it builds up, and that further suppresses inhibitory synaptic transmission. Okay? It's a very clear result. The problem was that multiple other research groups had already done this very same experiment with this very same drug and had reported no effect of inhibiting FAAH on inhibitory synaptic transmission and no evidence of tonic anandamide release in the hippocampus. Okay? Those are the experiments that turned out to be generated from tissue from male animals. And so when we do our experiments, apply the FAAH inhibitor in males, like everybody else has shown, there is no effect of inhibiting FAAH in slices from males. And this result is particularly salient in a way to me because anandamide has actually been implicated in a wide range of physiological functions, from responses to stress, pain, affective behaviors like anxiety and depression. And as a result of that, FAAH inhibitors have been developed as a therapeutic target by a number of different companies. And in fact, when we published this result, FAAH inhibitors already had been used in multiple different clinical trials, likely without any awareness that they could have differential effects in males versus females. Okay. All right, so now I'll just wind up by summarizing the things that I have tried to tell you today. In the first part of the talk, I showed you evidence for a latent sex difference in the mechanisms by which estradiol can acutely potentiate excitatory synapses in the hippocampus. And then I showed you evidence for a sex-specific mechanism by which estradiol acutely suppresses inhibitory synapses in the hippocampus, and a sex-specific effect of a FAAH inhibitor to acutely suppress inhibitory synapses. And I really want to try to hit home the point that I think the real significance of these findings is in the consequences that they may have for the development of therapeutics that target specific molecular pathways in the brain. You know, I study the hippocampus, and very often people are tempted to extrapolate our results to say that, to imagine they mean something about sex differences in behaviors that are engaged by the hippocampus. And I want to emphasize that we don't have any evidence that these differences are related to the types of behaviors that you associate with the hippocampus, but they're really more about making the basic biology that people like me study relevant to the translation and clinical findings, translational and clinical findings that people who use data like we generate need to have in order to design and interpret their clinical studies properly. Okay? And with that, I'll just acknowledge the members of my lab who did the experiments that I showed you today. All of the EPSC modulation was done by a graduate student named Anant Jain. And Guanji Huang and Nino Tabatadze are two postdocs in the lab who have been working on the inhibitory synaptic modulation. And with that, I will thank you for your attention. I'd be happy to answer the questions that you have.